Okay, so our last speaker for this morning is Jasna Brujic from New York University, and she will be talking about analysis and interpretation of single molecule force spectroscopy experiments.
Yes, so I think I'm going to be a little bit of an odd one out here because I'm going to be showing you lots of data, and I'm an experimental physicist, and so I'll try to show you how maximum likelihood algorithms and novel analysis techniques can help with a specific case study of looking at what the force spectroscopy of proteins can tell us about protein folding. So the motivation is sort of the grand question of what can we learn about how a protein folds, and how come that it always folds into a functional form, or almost always. And the interesting sort of background to this is that there are quite a number of diseases. You see, this paper is from 1995. There's already a list of diseases from cystic fibrosis to Parkinson's to mad cow disease, all of which can be pinpointed down to a single mutation in a given protein, and if that mutation happens, then you get the disease. And so it's very important to understand, if we can, what the mechanism for protein folding is, so that we can perhaps avoid these kind of errors. So that's the kind of broad motivation. The actual experiment that we do in the lab is really a Guantanamo Bay of proteins, in a sense that we pull on them, and we use actual mechanical forces to pull on them using very small cantilevers. We pull on single molecules, and then we study what the unfolding time distribution looks like, how that time distribution depends on the force with which you pull, and we try to test kinetic models and try to come up with some underlying principles of protein folding from that kind of data. And then we can, once we've tortured them enough, relax the force, let them go, and watch how they refold. And that turns out to be quite an asymmetric problem, in a sense that you see one type of physics for unfolding and another one for refolding, and I'll try to illustrate that. So what happens to a protein under force? My guess is the video now doesn't work, but perhaps most of you know what MD simulations look like. So imagine that these atoms here are jiggling around. This is ubiquitin. I chose that because it's a ubiquitous protein in every eukaryotic cell. It's the trash can of the cell, in a sense that it tags proteins that are misfolded and brings it to the garbage proteasome to be degraded. That's its role in biology. And you basically pull on it from the N and C termini, you attach it, and in MD simulations, the steered MD, you can actually learn a lot about what the intermediate structures are along the pathways, but the problem, of course, is timescales and complexity. This protein has 78 amino acids, so we're talking about at most, what is it, maybe a microsecond of simulation. Whereas in experiments we're talking about seconds for protein folding. So there's a big discrepancy between MD simulations and what goes in the experiments. And the simplest view of what's actually happening in the experiment in terms of a free energy landscape is to assume that there are two states, that there is a well-defined folded native state, and that there is a free energy barrier that you need to cross, and once you cross it, you go to the unfolded denatured state. And if this was the underlying physics, then you would expect to see single exponential kinetics for the unfolding times, and you would expect to see that if you increase the force, so the work done on the protein, so that's why it's force times distance, then the work that you put in should introduce a linear tilt into the landscape, lower this free energy barrier, and therefore you should see an exponential dependence also of the rate of unfolding on the force with which you pull. So there's two very simple Kramers and Bell model predictions to tell you what you should expect in the experiment if you assume that something as simple as this is going on. And 95% of all data in all of protein folding is analyzed using the Bell model and Kramer's for the analysis. So this is a little bit more detail of what the experiment looks like. You use some gold to actually deposit the protein. You need to be good at engineering protein molecules because you need to have tandem repeats. So these are clones of one another. There's 4 identical molecules that are covalently bound to each other, and the bacteria does very well at expressing clones. So I'll explain this is important for the analysis, why you need to have this, because you need a signature in the single molecule that you have reproducibility so that you know what you're measuring basically. And so you use a piezo and a cantilever to essentially pull on these guys, and then you can apply either constant force or constant velocity and measure the end-to-end length of this protein. So the downside of the experiment as compared to MD simulations is that you have only one single reaction coordinate. You're fairly poor in your data, and you can measure what happens over time. So this is a raw data trace. You've applied the force somewhere here, and you see that you see equally equal steps of the same length, which correspond to the unfolding of each one of these domains. And you can go and measure the time to the first to unfold, second, third, fourth, and fifth. And at the end of the experiment, there's a detachment of the cantilever. Yes?
So this is a force-controlled experiment.
Yes.
Why don't they all unfold?
Because it's a stochastic process. So even if it was a 2-state model with a single free energy barrier, you'd have an exponential distribution of times for pulling k out of n molecules, where n is the number in your chain and k is the kth one that you're looking at.
But so if you apply just one, but you have all this time, not force.
Yeah, this is just time, and indeed there is a trace that just shows here that you've applied a constant force at time 0. So that's the statistics you collect.
In your little cartoon, they all unfolded, then it detaches, but you don't know that.
Right, but that's because I'm going to build complexity into my problem. I'm starting, sometimes this does happen, other times we don't unfold all of them. So there are things we don't know in the experiment for which we need to account. Okay, you can repeat the experiment and you see the stochasticity of the process here. You have lots and lots of trajectories. You can measure each one of these dwell times. You can plot them as a function of the event number. And what you see is something quite surprising even already at this stage, which is that you have unfolding times that spread from milliseconds to 10 seconds. So it's a big span of unfolding times for exactly the same experimental conditions. And if this was Poisson statistics, you would not have such a wide spread of data. So what's going on? The first thing you can do is plot a cumulative distribution of these guys and take a look at, you normalize it according to the time window of your experiments, let's say 2 seconds, and then you can ask the question, okay, what can I fit this cumulative distribution with? And if it were Kramer's theorem would apply, then you would have fit a single exponential, but the take-home message here is that you need to fit a functional form for the unfolding probability, which is normalized to 1 at infinity and not your time window, and you're not really allowed to add an extra free parameter to normalize this distribution. You need to have a distribution where the unfolding probability that goes to 1 at infinity is consistent with the conditional probability on the time window that you do measure. So this is just a small a point because people don't do this in other groups, and so we wanted to make a point that those two things are not equivalent. Now this is where politics in science plays a big role, and I want to show you something funny. If you look through the data from and I'm implied in some of these graphs, but if you look from 2004, the first fit of the kind of data that I showed you is a single exponential fit, but admittedly there's very few data points, and that's why these trajectories look very staircase-like even in the cumulative distribution. Then you increase the statistics to thousands of data points, and what you see is that the single exponential essentially fits through one point on your cumulative distribution. Something else is clearly going on. Then you go further into the data, and the exact same data set from the same protein pulled at the same constant force is here fit with a log normal distribution. But the reason why they look so different is here is the cumulative distribution and here is the derivative. And then you have a fourth model in which in 2010 the same data is interpreted in terms of a static disorder case in the landscape, where you have a distribution of free energy barriers over which the proteins can unfold, and so this leads to non-exponential kinetics of this form. Yes?
So in the double log plot there, it seems that the exponent is decreasing as t going on, right? And since the bending out, is this generic or I mean?
So I believe that this is completely wrong, and I will try to show you that only one model can be at least closest to the data, if not right. There might be a fifth model, but I'm going to show you how you can distinguish between them. If you might say there's not much in it, why do you care between those four functional forms? Here I'll give some credit to all the authors on the previous slide, which is that if you plot a linear scale, all the four models that I just showed you, these three, the ones that are not the exponential, don't look so bad on the linear scale. And the only thing that's really very different is the time scale, which is the normalization factor, if you like, is different. So it matters for biologists because the time it takes for a protein to unfold is very different depending on which model you choose. But the chi-squared doesn't look so bad in any of these three models here. Now, if you zoom into what happens on at the short time scale here, then you can start to see consistent deviations. And here you see that between the four models, the stretched exponential distribution does the best job, because on this log scale, this is a straight line, and it seems to be the most consistent with the straight line fit. And the other models show deviations, but this is only at very short times. Okay? So if you start looking a little bit more closely, you start to see systematic agreement or disagreement with a given model. And the first step to doing that is to normalize the data correctly. The second thing you can do is you can test the robustness of that fit. If I take a time window of 1, 2, 3, 4, 5 seconds and I do the whole thing again, I should be at some point asymptoting to the same values of the fitting parameters. And that seems to be true for the exponential, even though the fits are ugly, the stretched exponential, but not so much for the Gaussian disorder nor the log normal. So the data set, as it's changed, the time window is changing, the fits are giving different results. So there's 3 orders of magnitude variation in the extremes, and again, now you need to have to start thinking about what's physical about this. If you have unfolding probabilities that are on the order of years, you know that that's not going to be true. So you can go to biologists and say, hey, what's the real time on which this protein unfolds? And you find that the unfolding time should be somewhere in the order of a second for this protein under these conditions. So we can sort of set up a kind of hierarchy of models and how to compare the data. One of the ways in which you can not filter the data according to the time window is what Eric mentioned to begin with, which is that you can take into account all the artifacts in the experiment using a maximum likelihood function. So I went to Eric and asked him, how can we not filter our data, include everything? And he said, well, you can just write down a probability to pick n proteins. This is n is the number of domains that are in a given chain. And then you have a binomial prefactor here that you need to basically weight the unfolding probabilities of each individual domain. So f of t1 up to f of tk is the probability to have unfolded by time t. And then you need to multiply that by the probability that there is a certain fraction of molecules that didn't unfold in the time of your experiment. So these are the unobserved events, if you like, which are weighted by, there's n minus k of those events. So you can basically write down an equation that will take into account the observed events, the chain length, and the unobserved events, and then you can look at this maximum likelihood function and see for which functional form of f of t do I get the most likely solution. So this was another way that we could test, rank the models against each other. This is an American slide because someone has to win and there has to be a kind of hooray moment. So these are robustness against filtering. Is it the most likely? Does it give reasonable unfolding times? And the exponential only wins on one side. The log normal doesn't do well on any of the three. The static disorder in an energy landscape, which turns out to be the same as whether you argue that there's Gaussian noise in the force due to experimental error, only does well in terms of the robustness against filtering, and the stretched exponential seems to win on all accounts. So we were happy to see that at least the three methods that we devised gave us the same answer. So if the shoe fits, you would think that's the end of the story, but the problem is a little bit that the stretched exponential doesn't have a very intuitive and interesting physics that we can really apply to the proteins, and it hasn't been published by others with NMR data or something else to support. Why would you see? Is there a anomalous diffusion? Is the protein subdiffusive? Is it because there's lots of different states? It opens more questions than it answers if it were to be true. So
how far is it from being a regular exponential? What's the
I'll show you that in just a sec. Next slide. So we decided to check, is this true for a bunch of different proteins, and how does the stretched exponential change with force? So this is your question, what happens to the exponent essentially. So these are two very different proteins. One is found in your cardiac muscle. It's the 27th immunoglobulin domain in your heart. And the ubiquitin we stay the same, and we try to see how persistent is the stretched exponential. If the stretched exponential is true, again, it should give a straight line on this particular scale. And so, for example, here you can see that for 5 different forces, these lines seem to be very nicely straight, and the rescaling seems to work well, and they seem to be quite parallel, which means that B seems to be constant as a function of force. Then we did the same for ubiquitin, and there the slopes start to get a little bit more slanted as you go to lower and lower forces, but the stretched exponential nevertheless persists. And you can see that here as well on the collapse. So if we put them side by side, the take-home message could be, 1 to infinity, all proteins are stretched exponential, and the exponent varies depending on the force. So let's see, what are the parameters, what do they look like?
Just to make sure that I understand correctly, so the lower the force, the larger the exponent B is. More
That's really my next, this is B as a function of force. So you see that in one case B is always constant, and in the other case, indeed, the lower the force with which you pull, the higher the constant. Meaning, if you were to get closer to 0 force, maybe it goes to exponential, and then biologists would be really happy who do wet chemistry and don't pull on them, and they could say all this behavior is just because you're torturing them. And if you didn't, then this would be 1. So that would be the way, if you wanted to publish this, you would need to say that. But I don't think that that's the whole story. because it seems to me like one of the things you can see here really clearly is the rate of unfolding is much, much higher for ubiquitin than I-27. It means ubiquitin is much, much weaker, which makes sense because I-27 is in the muscle, so it needs to be strong, so it's very resilient. So the energy barriers in I-27 are higher. So perhaps this data can be rescaled nicely onto energy barrier height, so that you would find that for the same energy barrier, as long as you have a high enough energy barrier, then these guys would all become at 0.8. And it's only because the barrier is small for ubiquitin, because it's such a weak molecule, that you end up seeing all these other pathways in the unfolding spectrum. That could be one qualitative explanation for what's going on. We don't have a theory at this point, so it's really just about saying we've observed something in which we have confidence that we've observed it, but we don't know how to explain it. One other kind of interesting observation is that if you do the inverse Laplace transform if you do a sum of single exponential processes and you assume that the distribution of rates is a Levy distribution, then you analytically get the stretched exponential to be the fitting function. So if you like the argument that each time you pull, the molecule goes over a different barrier, and that those barriers are for some reason giving you a Levy distribution of rates, then that would be consistent with what we observe. But it's not a very pleasing, it still doesn't give you a physical intuition for what's going on. And if you ask Eric, he would tell you that you need to look at the dynamics and write down a generalized Langevin equation and try to then get, what's it, the Hurst exponent that would somehow match with the stretched exponential. But that's another whole talk, which I don't have. So let me show you just a little bit where things are actually not working well at all, by the way. When you see people give talks in MD, you should always ask them, how well is this working in comparison to experiment? Because the experimental comparison is really not that good. You see, here is our data at 100 to 200 piconewtons. Here is what the simulators pull at up here. And if you try to bend over backwards to try to make the two match, then you've really got to draw this line, which the black one, which happens to be something called the cusp model. Basically assumes a two-state picture where you have a cubic function, I think, between the two states. And first of all, this is quite generous because of the way it's fitted. If you zoom in, neither data set really fits, but also the numbers that you get out are actually that the critical force is much, much higher than you would get in vivo. It's on the order of nanonewtons, and at least then you would get that the fitting parameters, which are extrapolated down to zero force, are somehow compatible with what they measure in biology. So the distance to the transition state is delta x, the free energy without the force, and this is the prefactor k naught. But this is just to tell you there's a lot of room for improvement in this gap and going beyond. So that's first part of my story. That's all we know about protein unfolding so far. The collapsing story is a little bit different to this one, because when you let go of the protein, intuitively you might say, oh, I'll just get a staircase backwards, and then I can compare the statistics of the folding times. But what you get is actually, I'm not going to motivate protein folding again. What you actually get is, here is what the graph that you asked for, which is that here is the constant force with which you pull and you see the staircases. Then you let go as low as you can, say 5 piconewtons, and then you pull again. And what you get is something very asymmetric, and you see that the protein collapses cooperatively between the domains. There's much more noise in the trajectories, and you're seeing that there's a very fast contraction at the end. So Am I kind of done? Is it?
No.
No, I have a little bit of-
35. So you have
Okay, so this part you know, and this is the part where you let go. See here I let one of the guys not unfold. This is the guy we didn't observe unfolding. And then here is where they're refolding, and you're still measuring this end-to-end length. And the thing that you should notice is that even though at the end of the experiment you reach the folded length, if you were to pull immediately after the experiment gets here, you'd get no staircases. They still haven't refolded. So refolding happens, unfortunately, in the place where we have no information, where the end-to-end length is the same, or within the noise of the experiment. So what we're investigating is what's the physics of hydrophobic collapse rather than folding. So it's a different problem in a way. You're going one way in the end landscape, you let go, you don't go via the same way back, you go via some other path that's the hydrophobic collapse, and then there might be a symmetry in what goes on here, but we don't see it. So this is really why
we can identify which one is unfolding which are. I mean, is realistic, this right picture, or?
no, we don't know which one is which. We only know the end-to-end length. We don't know. Okay, so as you can see, the slide improves in aesthetic quality. That's because this is Eric's slide, and this is to tell you that if the experiment were in equilibrium, and if you could get the unfolding and refolding to happen many, many times over the course of the experiment, as it does, for example, for RNA molecules, because they're very weak, so the barriers are on the order of kT, then you could start to analyze those trajectories simply by measuring what the end-to-end length, the histogram of end-to-end lengths is, and then doing a Boltzmann inversion to actually get what the free energy landscape looks like. And that works really well for very, very weak proteins and RNA, but there's almost none of those. I mean, proteins are much more rigid, particularly the ones that have a mechanical function. So we can't really use a simple Boltzmann inversion to try to get at the free energy landscape on the way down. So what people typically do is to pull at constant velocity instead of at constant force, so that you're doing continuous work on the system, and then you can use the Jarzynski or the Crookes theorem to relate the irreversible work, which you can measure, to the free energy of the system. And so the nice part about the Jarzynski identity and the Crookes fluctuation theorem is that there are exact expressions for this irreversible work and its relation to the free energy. The problem with this approach is that the amount of data you need is prohibitively large. I think also in almost all the papers where you will see this being used, but particularly in our case, because in our case we're doing instantaneous work, we're letting go at the time scale of the piezo. So we really don't have a hope in hell of using this method, and even in the cases where it has been used, the statistics are dubious. So there's a nice illustration of this problem by Liu and Grossberg, where they show that if you think about it as a piston, then if you let go of the piston infinitely fast, you need to track many, many, many rare particles that came with you in order to be able to reweight the distribution. And so the faster the piston moves, the worse these estimators become. And we're kind of in that category. So we didn't really know what to do with this data, and we didn't know how to interpret it. On top of that, it looks kind of ugly, so you can't sort of, if you had a nice staircase, then you might be able to say something qualitatively, but this was really hard to make an interpretation. And then I asked Eric, can you help out? And typically he has something clever to say when you ask him. And so he said, well, there's no free lunch. You can't say much unless you make another assumption. But if you make another assumption that the end-to-end length satisfies the overdamped diffusion equation, then you can actually start to make an alternative estimator for the free energy based on this assumption that you have a constant diffusion constant and that you have these pieces of out of equilibrium trajectories. Okay, so let me show you what that translates to in the experiment. Here is our experiment that's unfolded at the high force. We then let go of it. This is for your amusement. It reaches the folded length, and at that point the energy barrier is so high that it will never go back, and so you never see it flip-flopping between the two states. So what you need to do is reinitialize the trajectory at xu, then collect lots of trajectories from xu to xf and construct your non-equilibrium histogram of end-to-end lengths. And if you use this Langevin assumption, Chi Zhang, PhD student of Eric, and Eric came up with a closed form for the g of x based on this assumption. So here you see what it would have been had the trajectories been in equilibrium. You have the Boltzmann inversion, and here you have the correction factor, which is basically given to you by the derivative of the PDF, non-equilibrium PDF that you construct, and this reweighting that you do by reinitializing the trajectory. So it's like a trick. It's a clever trick to reweight what you measure to get it back into the equilibrium measurement. And so it's a neat tool because now we can go back to the data and we can measure this. And we can then estimate the g of x purely from the data. So how do we do that? We take this trajectory, we mark xu and xf in each experiment, we cut it, we then do many of them, we then put them in sequence as if they happened chronologically, if you like, and then we can just plot the histogram of these, and you see most of the data happens between 30 and 60 nanometers. So when you plot the histogram, what you get is this. The red data you see between 30 and 60 nanometers, that's when you have 3 domains in the trajectories. If you started off with 4, it's a little bit shifted, or 5 a little bit further. But the data seems to collapse quite nicely with a linear rescaling with n. So it seems to be that it doesn't matter how many, the physics doesn't change as a function of how many domains you have. And as a function of force, if you drop it at 10 piconewtons or if you drop it at 5 piconewtons, the rescaling seems to hold. Okay? So we were able at least to construct these non-equilibrium PDFs experimentally and then ask what does the corresponding free energy look like.
And the order in which you concatenate the data doesn't matter, is that right?
No, it doesn't, because it's just a histogram. And then you find something rather interesting, actually a few things. One of them is that the data rescaled by linear rescaling with n collapses all the data onto one response for a given force. And the other one is that a tilt by the Bell model between the two forces for the 5 piconewtons seems to do quite well in interpolating between the data. So it cares about force according to Bell, and the collapse seems to be cooperative between the domains. And now you might ask, so what happened to the diffusion constant? Because in order to understand the trajectories, you need to know both the time scale and the shape of the free energy. And so the classical way would be to do quadratic variation and estimate the diffusion constant, but again we can't do that because of the low pass filtering of the piezo. And there the method is very powerful because it allows you simply to look at the probability flux in order to find the diffusion constant simply from the actually you can see it here, by cutting the traces, so you go from x, you change xf to be at any given x along the trajectories, then you look at the probability flux of passing through them, and you can actually estimate d at every given point in time. So you can a posteriori check the assumption that the diffusion constant, which you assume to be constant, is constant. And what we find by doing that is that the diffusion constant doesn't vary very much as a function of the end-to-end length, and we find that it agrees very nicely with the estimate of the diffusion constant that's global by just plugging it back into the Langevin equation. And we find something very surprising, which is that this diffusion constant is extremely slow. 180 nanometers squared per second is an order of magnitude slower than an entropic recoil for any polypeptide, and it's 10 to the 7 orders of magnitude slower than what you would get estimated from MD simulations. So it seems like the hydrophobic collapse is actually having reasonable success in being modeled with this 1D Langevin assumption, but the diffusion is not at all what you would expect it to be just from molecular fluctuations. And here, if you look at just what the bead in water would be, you need a radius of 1 millimeter. And even if the cantilever is playing a role in damping the system, that should only be on the micron scale. So we think what we're measuring is all the internal degrees of freedom that the protein has that are being projected onto the end-to-end length, and that's what's slowing the system down so much and so crazily, and that's why we observe the collapse in seconds in the real experiment. So this is the experimental data. You can go back and check the theory by generating trajectories using the g of x and the d that you estimated, and this is really beautiful because you can see that the shape of the trajectories, the noise, the times, and so forth really agree very nicely. And so again, you have some kind of a validation of the procedure that you've applied, despite the fact that you know that at some level it has to be wrong or incomplete. And one other kind of interesting if I were to ask you before I started this talk, why is this thing diffusing for so long and then collapsing cooperatively, you may have said, because there's a little barrier somewhere and it's hovering behind it, and then it collapses as it crosses. But if that were true, then there would be an exponential dependence of the time on the number of domains. And the fact that the number of domains doesn't seem to affect the collapse time very much is consistent with this hovering and diffusing and being cool on this flat landscape. So we think we've discovered really an alternative mechanism for the hydrophobic collapse, which is consistent with a very so this is with a physical model which is based on Pierre-Gilles Degennes' expanding sausage model, being sort of agent provocateur in this naming game, but basically the idea is that the polypeptide with which you start is coarsening over time, and the blob size or the globule size in the sausage is increasing, but the volume of the sausage is remaining the same. And so what you end up with is going from something that's taut into something that's a sphere with a given volume. And well, this is too blurry to go into detail, but this is basically the worm-like chain model for the entropic collapse, which initially takes place. Then there is the linear tilt with a force, the Bell model, and the sausage model is simply this relation between the blob size and the volume that you end up with at the end. And we get numbers that are very reasonable, on the order of a few amino acids for the blobbing process. I don't believe that this is a theory or a real understanding of what's going on, but it gives a sort of mean field hand-wavy argument for how this polypeptide is really first collapsing, excluding the water, and then folding. So there's the expanding sausage, and the surprising result is that the diffusion constant has such a small value, and it sort of starts to ask questions about, again, what don't we see in the experiments. And I just wanted to kind of conclude by saying that in the first part there's a non-Markov end-to-end length coordinate that we see through this stretched exponential kinetics that we measure. So other degrees of freedom do play a really important role in what we measure, whereas in the collapse dynamics, simple diffusion seems to be adequate, if not explaining what the actual physics is. So here are the people in my group. Frauke Grete's group did the MD simulations that I showed you to try to extrapolate between the data, and Eric and his student were the main actors in the theory part. So thank you.
Thank you. I have a few questions, which is fine.
So, I mean, you mentioned that in the stretching and the collapsing, the dynamics might be different, but since you have this free energy from the collapsing, I wonder what will happen if we use that free energy and to learn the dynamics in the stretching part.
In the stretching part. So I think it's really two different parts of the energy landscape. In one case, you're very highly taut. All the side chains are exposed to water, and so you're at something like 85% of the contour length to begin with. So nobody sees anybody else. And so the first physics that then, instead of going down the path which would lead to the independent domains refolding and clustering together, you seem to go down a path which is somehow to first exclude all the water, make them go into this blob, and then they sort themselves out in a way that we don't know yet. Is your question, why don't they?
You mean the free energy will be completely different for these?
I think it's completely different. In the other case, you start off where the native contacts are already there, and you have this viscoelastic object that has its breathing modes and really restricted dynamics, and then you're asking who's the weakest link when you pull on it that will lead to a global destruction and unfolding. So it's really, one is about kind of waiting for that rare event where something unzips the protein completely and gives rise to each individual step. And the other case, you're asking what's the dynamics of water exclusion in the whole chain.
You can see this process by MD. In the sense you can see the first going, because I am astonished. The excluded volume of the water should
You can do MD simulation of polypeptide, decaalanine, for example, in implicit solvent. You can kill yourself putting explicit solvent, but nothing as big as that has ever been seen to collapse in an MD.
You cannot put yourself in a state in which the collapse starts fast, and you see, because I think that the water should be expelled together with the contraction.
If you do the MD and you start, you
In a
pull
very
your pipette
probable-
in, it's a huge box of water
Yes.
that you need to have, and it will just over there, and you will never see any of it. It's quite tricky actually. This is beyond the time scale of MD.
But
In the other
steered MD simulations see
Yeah, you
folding.
can do steered MD.
If you do steered MD and you just copy the experiment, you can see the whole collapse.
And then you can see in which way the contraction is accompanied by the expelling of the water.
Yes, but
And you can see
yes,
they are
but
the same scale or not.
yeah, but just to give, if you do the experiment, right? For example, when you pull is 1 nanometer per second or something like that is being made, right? Whereas in the
400,
you
but
pull at 1 meter per second. Otherwise,
Yeah.
you don't see it.
It's
That's
between
the
1 meter
explanation of
and
this
one-
graph where you see pointed at 7 order of magnitude difference.
in time scale. Yeah.
Ah, okay. So in reality, even the fastest scale is low. And then everything is
For example, Dave Trumalai has done Go model simulations where you use coarse grain models and you do see the hydrophobic collapse of the entire chain, but there's so many assumptions in the model that are tuned by the experiment that you don't really know.
No, but now I was reflecting the argument. What is faster for you experimentally, from the experimental point of view, is very slow for the microscopic dynamics.
Mhm.
And so the fact that you have 2 times scale is consistent with the fact that they can happen one after the other,
Yes.
because they are slow, so there is time microscopically to feel.
Yes, but I think it's biologically very relevant. When you go shopping and you pick up your bags and you put a force on your muscles, you can calculate actually on your muscle what's the force per unit domain of your muscles, and it's on the order of 200 piconewtons or so, and it makes sense that when you finish doing your exercise or your carrying, you're basically first collapsing. You get some length out by the initial contraction, which is sort of entropic in origin, and then you sort yourself out to have your molecular mechanical machine working. You have more time later on. So I think this is more like the fast way that you get length out to get the reversibility.
Yeah, my question was naive, is that when you say fast on this macroscopic scale,
Yes.
but on the microscopic of the molecular dynamics, it is very slow.
It's
So
very slow,
it's all
exactly.
the time to arrange so that that's possible.
In the experiment,
Exactly.
nothing below a millisecond can be measured as the
Absolutely.
fastest time scale, and nobody can do ND up to 1 millisecond. So
But the
there's a
water can
total
do a lot
gap.
of things.
They
to
don't
arrange.
talk to one another.
Yeah, you go fast, but again slow.
Yeah.
Okay.
I think people are building microsecond AFMs, and you will see that when that gap gets filled with data, you'll see that they're actually not compatible with each other. There's just different models to fit them.
Your Boltzmann inversion was non-parametric, is that, I understood that correctly?
Yes.
But then you wrote down the free energy that was parametrized. Was it,
Yeah,
did you
the
fit that to the non-parametric free energy that came from the Langevin, or was that right
here is the.
separately?
Oh no, wait, I forgot. Here, I have to click. This is the Boltzmann inversion that you end up with.
Well, and what's the connection between that and the parametrized G, which you wrote down? Is it the end you parameterized?
Sorry. Here, this is the one. This is the one where we actually measure the row of x. We measure the row prime of xf, and we can actually do the reweighting of. So we take the data and we estimate what the g of x will be given this measurement. Is that what you mean by parameterized with the end-to-end length?
That's non-parameterized.
Yeah.
But then at the end of the talk, you wrote down an expression for the free energy that was had some particular functional form.
Oh, okay, I see
What's the
what you
connection
mean.
between that
I see
and
what
this?
you mean. Here it's just in terms of the end-to-end length. In the other case, it's to relate the end-to-end length to this collapsing model and having information about the origin of the shape of the g of x in terms of the size of the blob, the persistence length of the chain, the contour length of the chain. So this is like going from, this will just give you what the shape is without you knowing the physics at all, and the other one is trying to find a functional form that fits this shape in terms of the parameters of your system, and to explain it. And so in the other case, we try to relate it to this blob size in order to make an argument that you have a cylinder sausage, which is basically getting fatter and smaller because of some coarsening.
Is that a good fit? So if you take your other fit
Yes,
to that model and draw from it,
yes.
do you get curves that also look
God,
like
I now
the,
understand your question entirely. And that's the difference between, I don't know if you can tell, which is a good thing, black line is the fit, and the colored lines are the G of X from the data without the. So
it's a good fit.
it's a good fit, but actually we lie a little bit, because if you look at the black one, it's consistently sharper than the data. The data is always a little, the way it's drawn, it looks better than if you zoomed into that last leg. And I think that that's because it's not as cooperative as the physical model would predict. It's something more consecutive, that there is a sequence that matters.
That's when it's fast.
Yeah, the estimator is the poorest also there, because it's really fast, so there's very few data points.
How many of these collapse trajectories?
That's so the remarkable thing is that you need like 15 trajectories, 10 to 20 trajectories, instead of, we thought we might need 1,000 or 10,000, and then all of this would go to water, because it's 3 years of PhD to get 200 trajectories. So there's a kind of a grant problem with how well the estimator works. But it turned out that with this assumption, you don't, with the simulations, you found that it was incredibly low, 10, on the order of 10 trajectories.
Is there any advantage in having more of these molecules hooked up together? Because you have the ND3, 4, 5, so that's the number of copies of the protein that are sort of put together, right?
Yes.
So is there any advantage in taking more than?
Not in terms of this, because you just have a linear descaling where you do G of X divided by N, and then it's really those trajectories that you then use to, you can estimate, depending on which assumption you're trying to test. We first separated them, showed that they do collapse as a function of N, and then you can use all of them, pull them together and say, can I get a more accurate estimate for the global collapse?
Okay.
Cool.
There are no more questions. Let's thank all the speakers of this morning's session.